Oh, we're really worried, Damon. So do let me introduce you before you get started. So when you're ready, I'll do that.
Go for it.
It's 7 o'clock. Let's go.
Okay. So welcome, everybody. I'm Terry Smith from the Pollinator Posse. I hope a number of you got to meet Victoria the corpse flower in real life, but I know Damon's got some fabulous pictures of it. If you didn't, so you'll get a little bit of it vicariously. I want to introduce Damon. We're so grateful to him for stepping up to do this tonight. We'd hoped to do it before the corpse flower bloom, but it surprised us. So this was the best we could do, but he really rallied and got his talk together. So Damon was born in Klamath Falls, Oregon, and raised in Calaveras County. He attended St. Mary's College in Moraga, so not far from here, where he worked on the local newspaper while earning a biology chemistry degree. He taught high school in Portland, Oregon, and moved back to the Bay Area to work on the Human Genome Project at the National Labs Joint Genome Institute. He spent a bit of time pursuing an MFA in natural history and science filmmaking in Montana. We'll see some of the benefits of that visual training, but returned to Oakland to work on biofuels and single-cell genomics. He currently manages an apartment complex in downtown Oakland because he didn't have enough to do. And he works for Biorad Laboratories, designing curriculum and training educators from Colorado to the coast in biotech, and is well-versed in local fungi, plants, and slowly but surely the critters of Lake Merritt. You didn't even mention this, your barcode, the lake of Lake Merritt. You might talk about that a little bit at the end. Okay. People be interested in that project. So with that, I'm going to turn it over to Damon.
All right, thank you, Terry. And I want to say, first of all, thanks to also Kai and Susan for opening up their plant to everybody to come see. I mean, that's what initiated all of this was, oh my God, we got a corpse flower and everyone's gonna get to come see it. Well, we're gonna plant all the stuff around it. Oh my God, she's coming early. Okay, well, we'll have a post celebration instead of a marketing PR event about getting information out. So that's where this presentation came from. And so I thought I was gonna have maybe another week and a half, 2 weeks to work on this. And then we got the call Monday morning. They're like, oh, it's happening. And then I was like, okay, I think I can put together something by Friday. So this has all been compiled by Friday. So just FYI. In the chat, what I'm gonna do is I'm gonna drop the link to the slide deck. And that is there mainly because in my slide notes, if you're a super nerd, there are the papers that I'm using to come up with the conclusions that I have in some of the slides. So that way you can fact check me, which is always important, right? So know that that is there. So with that being said, I'm gonna share my screen and we'll jump into the talk. Oops. Screen.
So if you haven't muted yourself, could you do that?
I'll go through and mute. I'll mute everyone so they don't have to worry. They can just
then, okay.
And then I'll ask them to unmute at the end.
And actually, Terry, can you make sure that the recording's still going on? I just got a weird message there briefly. Actually, let me stop the screen. Sorry, guys.
It says it is recording on my screen.
Okay.
It's
recording. So I think you're fine.
So Terry just shared with me this photograph, which is Victoria in the Danois portion of the celebration on her way.
Damon, you're freezing.
Down. But this is the whole reason that we're and fly. Give me 1 second to try and start this share again. My computer was the fan kicked on. I think it's just because the sun was just beating on me here.
Okay.
Let me do this one more time. We'll do the screen share.
And your audio is going in and out too.
Yeah.
Okay. How about now? Is it a little more stable?
Yes, looks better.
Okay. So I think part of it is when you try to pull up a giant photo heavy slide deck, the Dell computers are like, oh my God, what are you doing to me? So now that it's up, we should be good to go. So in tonight's talk, I'm going to do an overview of Victoria and corpse flower biology, kind of a high level skim of the life cycle, the chemistry, and a lot of what we don't know. And one of the major things about corpse flowers is that they attract some unique pollinators. And so we're going to do a dive into the world of Diptera, flies. And then we're going to branch out and connect this to our local ecology by talking about California native plants that are pollinated by flies. So to kick things off, I just want to say thank you once again for letting everybody come see Victoria. So this was the collage that literally got cobbled together by me just asking people on social media, hey, would you send me your photographs of you with Victoria? And they just came roaring in. What I love about this is just all of the different facial expressions of people interacting with Victoria. Everything from the bottom left-hand corner there, which is like, oh my God, this smells so bad, to these looks of surprise, enjoyment, things like that. So this event, thank you guys, it creates so much kind of joy and excitement in the community. Victoria was spectacular. So when she blooms again, get it on your calendar and get there to see her. So these are a couple of photographs that Kai let me get from different angles using a stepladder and all kinds of things so that we can see her in her full grand glory. An enormously huge bloom, beautifully colored, and just stunningly lit too within that greenhouse space. So what are corpse flowers? Well, corpse flower name is actually a direct translation from the Indonesian name for it. And so these are originally from Sumatra, where you have a lot of Indonesian speakers. So the traditional name for this is Bunga Bakai, which generally translates into dead or smelly corpse flower. Amorphophallus titanum, if we go with the loose Latin of this, this more or less means a really big, giant, deformed penis, which if you look at the spadix, okay, I can see how the Latin people would get to that. It's the largest flower in the world with an asterisk on it, of course, because biology always comes with asterisks, but we're just going to say largest flower in the world. It's endangered in the wild, and it is in the family Ericaceae, which we do have representatives of here in California, which is always kind of one of the cool things if you're getting into plants is start learning families, because you get to see these giant connections across the globe of what plants are related to what. And so close home to us, the most notable ones are skunk cabbages, which actually kind of have the same shape as the corpse flower. And then one that actually kind of blew my mind, I didn't know that was in the same family, which is duckweed, which you will find everywhere in ponds all across California. And it is by far one of the smallest plants you'll ever see. So it just did not even show up on my radar that this would be in the same family, but we'll dig into those a little bit later. So the distribution of Amorphophallus as a genus is predominantly in the tropical and kind of semi-tropics from Africa to Southeast Asia. And it's a relatively large genera. For Amorphophallus titanum, though, it is only in Sumatra. And it's actually in just kind of a few locations there, and it's becoming relatively rare to find in the wild. If this kind of look into corpse flower and Amorphophallus gets you stimulated to be interested in the botany of Sumatra, I'm just going to throw it out there. There is this fantastic conservation biologist in Sumatra who I interact with a couple times a year, Punky Nanan Natama Pratama, who does a great job of keeping you up to date of what he's seen in the field, which also includes a lot of Amorphophallus. And so there's two of them right there. And that middle one, geez, I would love to see that in person sometime. But somebody just to be aware of, you can easily follow on Facebook. He does a lot of great projects involving camera traps and then also reintroduction of plants into the habitat and a lot of education with local young children in Sumatra, because they are the ones, right, that are going to be responsible for making sure these forests stay healthy going forward. So life cycle. This plant has a really long life cycle, which if you were sitting in on the social hour, you probably kind of gleamed from hearing that from mama to the new plant was on the order of 16 years, or at least the new flowering plant. So in this cycle, let's go ahead and just start with the seed. When you get the seeds and they find a place to germinate and get going, you can kind of jump into two potential cycles down the road. The first cycle it'll jump into is basically the leaf cycle, where you'll get this corum that starts being fed by a large shoot of leaves that gets produced, and they are supposed to hang around for about a year and then they'll recollapse. All the energy that they're picking up during this time is getting stored in carbohydrates that are going into this large corum underground. In fact, so much so that one of the Eumorphophallus, actually that one I showed you in the last slide in the far left corner, is actually used as a food source. People will grow that for the corum and use that as a food source throughout, I think it was Africa and Southeast Asia. It's now being used as a food source. So, but basically you go through this leaf cycle for many, many years until you build up enough kind of stored energy underground that the plant can initiate the flower cycle. Because if you're going to produce the world's biggest flower, you got to have a lot of energy to do this with. And so these corums can get really, really large. I think some of the upper end estimates of these are 250 pounds. But once you've kind of hit that cycle where it's ready to bloom, what will take place is you're going through the back into the major corum, and then you'll get the spike starts to emerge, and then the spadix comes out, and then you basically get a flowering that takes place over about a 2-day period, and that's it. You're done. All that effort, all those many years, just to do 2 days. And so these are really important 2 days because this is your one chance as this plant to get frisky with other genetics in your area, i.e. pollination. And so we'll basically have cycling of male and female flower parts so that we don't self-pollinate. And then once that pollination has happened, kind of collapses, and it takes a while for those fruit to then be produced. In fact, another look at this is with this graphic here that I stole from somebody's very nice blog about, I think this person's out in the Bronx. So these spadix on these flowers can be up to 10 feet. The spathe, which is actually a leaf basically encapsulated, and like I said, those corums can get up to 250 pounds. But what I really wanted from this guy's graphics is this nice diagram of where the flowers are. So when we come to the bottom of the spadix, what we find is we find a band of male flowers and then a band of female flowers. And these do not occur at the same time. They emerge at different days. And that is so that you don't self-pollinate. And then those seeds take anywhere from 6 to 12 months to fully mature. And then they are, for the most part, distributed via birds, especially hornbills will take these because they're brightly colored. They're kind of this orangish red color. They supposedly have like a little bit of a sweet exterior to them. So the hornbills will take them, chew on them, and then drop them. And this is how you get distribution of the seed. So, man, when I thought there was just one cool organism involved in this, you've got 2 fantastic Southeast Asian organisms involved in moving this stuff around. So this is a couple screen grabs from a fantastic time lapse the Chicago Botanical Garden has on YouTube, just so that you can get a feel for what the flowering period looks like. So I went through their video and tried to count their day cycles based on when lights came on. And so the emergence of the spadix took about 12 days in their case. And then once it kind of hits its peak, you've got about a 3-day period of more or less what I'd consider more or less the flowering to the denouement. So you've got 1 day of female flowers being available, close it up, or the flowers close up. And then the second day, basically the male flowers become available. And then after that, you have kind of post-coitus, post-pollination event, close for business, and it tips over. And that's where you get seed formation. One thing that would be surprising is if we could see an infrared, because what we would see really surprisingly with this plant is it produces a lot of heat. So especially during the night cycle, which is important because if you were trying to get the pollinators that this is using, which is predominantly, we think, flies and carrion beetles, you are wanting to convince these pollinators that you are a dead and dying animal that you should come visit and maybe lay your eggs in. And so having a little bit of warmth helps. And also that warmth at night is going to help you broadcast a lot of these volatile compounds that a lot of you guys got to experience in person, those stinky smells. And so there's some really cool studies out there where they monitored the heat of this over the 2-day cycle. And what you see is that it peaks, you know, relatively late at night. So on the first night, when you have the female parts coming out, you reach kind of the highest temperature. And so for us Americans not used to centigrade, 38 centigrade is you're getting up into like, you know, a bad day in Sacramento. like 106, 110 degrees Fahrenheit. Drops down during the daylight hours, and then once again picks up for the male cycle the next night. And so this is your best bet at kind of making sure that beetles flying at night and flies flying at night find you. Give yourself a warm beacon and a high volatilization of those compounds. The cool thing is some of these IR photographs they had from this paper, you can actually see that it even goes one step further. So the top of the spadix is, of course, giving you that beacon effect of like putting out a bunch of heat. But then if you go down where the business units are, the flowering, heat will occur behind which flowers are active. And so in this case, that bottom photograph, you can see the male flowers are active there, and it's just pumping out heat behind them, which means drawing those insects in because of the warmth and because of the volatile components being further aerosolized into the wind column right from those. So in my head, I'm like, holy crap, how does this happen? How does a plant generate this much freaking heat? And luckily, somebody in 2019 had that same question and a lot more funding than I do. And so they did what we call a transcriptional analysis of the different tissues of a blooming corpse flower. So this means you basically take out the different tissues of the corpse flower as things are progressing, you grind it up, and you take out a compound that everybody's familiar with now because of the pandemic, RNA, which is basically the message from your DNA about what to produce. And so if you look at RNA, and you can count how much is there from different places on the genome, this tells you about which pathways the plant is basically driving really hard, more or less has turned on. So in this paper, they had two pathways that were turned on, just amplified to the point where they're producing heat probably. The primary one being the MEP pathway, which ultimately takes a lot of that energy you've got in the corum and ends up turning it into terpenoids. And along the process, you get a lot of these intermediate compounds that end up being the compounds or being ones you can get transformed into some of the compounds that you can find in the headspace, basically the smelly air volume from these different tissues. Somebody else did a separate study on looking at just the headspace around different tissues from the plant. This blows my mind. The different tissues are producing very different signatures when it comes to the volatile compounds. So when you look at the appendix, and this is another word for more or less the bottom part of the spadix, you have a whole series of stuff that's radically different than when the male flowers are out and is radically different than when the female flowers are out. And then the spade is just all the time producing butyl acetate, which is one of those things that you associate with rotting dead stuff, which is just amazing. To highlight a couple of these compounds, just to kind of give you a frame of reference for where they belong in your nose or maybe not in your nose. We've got dimethyl trisulfide, dimethyl sulfide, methylthiol acetate, trimethylamine, isovaleric acid. I know those are kind of a tongue twister for a lot of us, but anybody that's into organic chemistry, these are ones you probably know pretty well. All five of these ones are really well known from stinky cheeses. And they also impart some of the flavor that some of us enjoy or don't enjoy in certain cheeses. Dimethyl trisulfide is the major component of Limburger cheese that gives you that Limburger-esque-ness to it. Those top three ones, if you look at the organic chemistry structures there, you'll see they have sulfur in them, the S compound. These are found all the time in stanky beer. So if you had a beer and it went bad, these are the compounds that you will find in that beer. These compounds also are what you get when you cook a lot of brassicas and allium. So when I say brassica, we're talking anything from like cabbage to broccoli and everything in between, because like half the vegetables we eat are brassica, just in a different form or another. Allium, of course, means onion, garlic, those type of things. If we move down this list a little bit, when we get to methylthiol acetate, this is the primary scent of kind of stinky, starting to get a little too ripe fruit. We also pick up a little bit of this from isovaleric acid, and this name derives from the valerian root, which you've ever worked with, is kind of stinky, it is kind of funky tasting. So both of those compounds are involved with that ripe fruit scent. So if you're by Victoria, occasionally you would get this where you're like, ooh, that's not too bad. I am kind of almost intrigued by that scent. And then the rest of it would follow through and you'd be like, and now I'm stepping away again. The trimethylamine is the rotten fish odor that you may have detected as well. It is one that we actually produce as humans a lot of times because of our gut microbes. So if you eat a lot of food that has a high level of like choline in it, your gut microflora will transform that into trimethylamine, which really easily goes into your blood system. And all of a sudden you sweat a little bit of BO that's a little more fishy than everybody else. The isoflavonoic acid, which has a little bit of little aspect of rotten fish, a lot of cheese, and a little bit of ripe fruit to it. This is the primary flavor that you get from Brettanomyces fermentation. So, a lot of you Bay Area folks and, wine aficionados, you'll recognize this as the essence of leather, or if you're really writing up a wine bottle, it's like the essence of an English saddle for a warm riding night in your wine. However, if you go a little too far with the Brettanomyces fermentation, you get unruly barnyard, which is what us beer aficionados appreciate in our saisons. And so that gives you a little bit of a picture of some of that organic chemistry that's going on with Victoria, the corpse flower. So why are all those compounds being produced? Who's it attracting? It's not attracting winos and beer aficionados. It's got to be attracting other types of pollinators. Unbelievably, after lots of searching on a very, very long Tuesday night, I could not find a darn paper that did a cohesive list of pollinators, which kind of blows my mind. So around like 3 a.m. in the morning, I'm just like, oh my God, why am I still awake trying to find this paper? And I find on the Kew Garden site that even they recognize that there is no comprehensive study of pollinators for the corpse flower, which kind of blows my mind. But then if we also think about the way that some science gets done, it doesn't blow my mind, right? When it comes to a lot of plants, we're very focused on crops. What things are going to influence the crops, because that's how they get funding. But there's a lot of people that do speculation about, what's growing or what is doing the pollination here. So because those compounds, and because of the obvious smell of it, and just from kind of people looking at it in the field, but not a comprehensive study, we can say carrion flies and carrion beetles are definitely involved in corpse flower pollination. And so if I do a lackluster study of this through iNaturalist, I can go to Sumatra and I can look at the carrion flies and the carrion beetles that people find near the habitats where the corpse flower blooms. And these are the ones that are on iNat right away. And if I dig in even further, which means I find pictures of corpse flowers blooming in the wild, and I'm zooming in on iNat to see what's on the spathe, I actually do find the carrion flies. I can't find the carrion beetles because they are probably trapped down on the bottom there, but I do definitely get evidence that these carrion flies are involved. So if anybody wants a low-hanging fruit, masters, this would be a really fun one to do, just throwing it out there. And because if you got this, like half of those journals would love to put a giant corpse flower on their cover. You would get great press and probably a really fun 2 years in Southeast Asia. So with that, let's talk about some of these pollinators then. I'm going to leave out the beetles because beetles do get a lot of love because they're really shiny and pretty and they've gotten a lot of funding studying all over the time. But flies really get the short end of the staff. And so we're going to talk about flies not being poopy pollinators, even though we do find a lot on poop. They are really great pollinators. In fact, many times in the gardens at Lake Merritt, you will find flies on flowers completely dusted in pollen, like these 3 individuals. And just to put it out there, I'm giving a great thanks to Constance Taylor, who allowed me to steal a lot of her slides from her recent fly talk. So some of these are things I've clipped from hers. So after bees, a relatively new study out of 2019 suggests that flies are likely the biggest pollinators on the planet if we're talking about an insect order. So following the money, right? Because that's the way some research gets done. Nobody's going out there and surveying a whole bunch of native plants yet, but if we're surveying crops in the United States, after bees, the big ones that show up are here on the right. So hoverflies, blowflies, houseflies. You add those up, you stack them under the order Diptera, which all flies are in, and it's pretty obvious flies are the second most important pollinator in the United States. And playing upon that food piece, there are a whole bunch of foods that you can't enjoy if it wasn't for the flies. And so all you chocolate lovers, get down with loving flies, because if you don't have flies, you do not have chocolate. Simple as that. See that little tiny guy up there in the corner? That is who you've got to thank for chocolate plants being able to mature and produce that thing that you love. So hug them, well, don't squeeze them too hard. They're pretty small. They probably will die. but we need to learn to appreciate that flies do a lot of pollination for us. And so after chocolate, we do have a lot of crops in the United States that get their pollination done by flies. Even if flies aren't the dominant ones, they are still an important pollinator within getting these fruits to market. So we're talking cherries, apricots, mangoes, blackberries, raspberries, basically anything in Rubus, peppers, carrots, pawpaw. Geez, I wish we could get pawpaw on the West Coast. Black pepper, strawberries, apples, cilantro, onions. And so there is a lot to be thankful for when it comes to fly pollination, just from a, I'm a human, I like to put tasty things in my mouth point of view. In fact, so much so that now that humans are starting to get aware of how important flies are, we're actually seeing legitimate studies, mainly agricultural focus, getting kickstarted. So in 2019, New Zealand started a number of really interesting studies using drone flies to pollinate crops. And so what they're trying to do is build habitats around large crops that will encourage basically this drone fly to show up in mass numbers and hopefully be better at pollinating those fields than the bees that they're using currently are. And also anybody that's living under a rock, you may not be aware that a lot of European honeybees are also going through a lot of problems with disease and big die-offs and things like that. So from an agricultural point of view, it's really important to A, recognize that flies are doing a lot of pollination, B, figure out how to encourage habitats where flies can exist near crops to help our crops survive. And then amazing to me, part of the reason I wanted to give this talk was to actually be like, okay, at the end of this talk, I want some people to think about how can we generate more fly-friendly habitats in our gardens and maybe get a little working group around that. In 2017, a Spanish consumer product actually hit the market where I think they've got 4 or 5 different species of flies, and they tell you which crops they work best on. And you can literally, if you're in the UK or in the European Union, you can order this and it shows up at your door. It's just kind of like how ladybugs are used in the United States. You get a little container that's got the larvae that you need. You place that in the garden, and all of a sudden now you've got queen flies or other types of drone flies flying around your yard doing pollination. And the other side of this is, I know people are like, but flies, you don't get honey out of them. So, I mean, cool, but you know, I kind of like honey. I like getting the secondary benefit of having that pollinator around. Well, flies, you get some other benefits, even though it's not sweet stuff we can put in our mouth. A lot of larvae, especially from hoverflies, are predators of aphids. And so you get this thing that's going to pollinate your plants and is going to help you with pest management, which is awesome. The other beauty about a lot of flies is they have a much simpler and easy to facilitate life cycle than bees do. And so that's why I think New Zealand's kind of really doubling down on some of these studies right now, is they also think it might be just easier management practices in the long run. Why have we not been doing this previously? Well, you know, PR. Flies have a really bad PR company working for them, and so they just have not got the press that they need. And so we're hoping to give a little bit of that to them today. So let's talk about flies in general then, because they're so important in pollination. We at least got to understand, get our heads wrapped around about why they might be such good pollinators. Well, there's a ton of them. There's about 150,000 species of identified Diptera around the planet. And that is the order that flies are in, which basically equivalates to about 14% of known insect species are in the order of Diptera. And it ranges everything from crane flies to drain flies. So the drain fly is that one up on the left-hand corner there. And if you guys have not seen one of these and you live in an urban habitat, you're just not looking hard enough. Or you may have confused them and thought that they were a really weird, pretty moth. They're actually a fly. Down beneath that, we have the world's sexiest mosquito. Nice with, like, you know, shiny silvery blue leggings. Yes, mosquitoes are flies. I know, and people are like, oh, that ruins it for me. Yeah, mosquitoes do pollination as well, but they also like to feed on us. So yeah, I feel you there. We also have things like little-headed flies, that top right-hand corner one, and signal flies. Both of those, the top one looks kind of funny and cartoony, and we can all love that one. That bottom one looks scary as all heck, but don't worry, it's not a fly with a gas mask. It's just a really weird feeding apparatus. It doesn't bite you. In fact, no flies really have chewable mouth parts. It's mainly a giant kind of orifice for it to land on things, spit out enzymes, and then suck them back up. The way you tell you've got a fly, because there's such a big diversity of them, I'm going to break it into just a handful of features. And these aren't hard and fast per se, but it gives you the idea that you're in the ballpark of a fly. So these features, not bugs, haha, tech joke, are as of this. Di means two, pteron means wing. So Diptera are these two-winged insects. And the two wings actually refer to two sets of wings. So we usually have two dominant wings that are visible on the back of most flies. And then the second set is actually these wings that have decided to do something else. They've become halteres. And halteres are a really, really cool device that flies have that are basically gyroscopes. So you know when a fly is flying around and you swat at it and you're like, I'm a human, I should be much better than any other animal on the planet, you know, I'm much more evolved, whatever. And the fly still outdoes you all the time because that's not really the way things work. Evolution doesn't work in, you know, archaic things like that. It's because they have these halteres that allow them just to navigate things really, really well. And they also have really short nervous systems, which means they're going to outperform you every time in a motor skill. So just learn to own that. The other portion about flies, they don't have chewing mouth parts. They either have a stab and suck system, which is your mosquito, or they have the spit and suck system. So you throw out enzymes onto the thing you're interested in. Maybe it's poop, maybe it's some pollen, and then you suck it back up with that weird mouth part that hangs out. Some flies, you can really see that really well. Other ones, you have to get in there with a magnifying glass like Drosophila. You can see it, but you got to get under a microscope to see it with. The other attributes that make it easy to kind of identify, oh, I've probably got a fly here, are really big eyes and then really short antennae on the tops of them. Some notable Diptera families, and this is for those of you that are interested in pollinators. If I go into the Lake Merritt Gardens and I'm looking at the flowers there, these are the ones I most commonly see on our flowers or in the garden. We've got our hoverflies, which can easily be mistaken as a native bee if you're not getting up close and getting a good look at them, because they actually kind of color code themselves so that they're like, hey, maybe don't mess with me. Bee flies, which are super fuzzy and cute. And then we have carrion flies, which we mainly find in Lake Merritt because they are going after those fungi we were talking about before, the lattice cage stinkhorns, and then tachinid flies. And those carrion flies too will actually end up on a lot of flowers that if you get in there with your nose, you might go, ooh, I was kind of hoping for something good, and that's not good. But these are ones to just be aware of. I'm not going through the whole list of the Diptera families because there's 150 of them. I don't have that type of time. You don't have that type of time tonight. But there's this thing called the internet. You can just go on a deep dive after we're done. Okay. So then after flies, I was like, all right, the big show in here was to connect Victoria to our local plants that are fly pollinated. And in the back of my head, I was like, this is going to be easy because I'll just go online. I'll find things in the same family as a corpse flower. I'll find papers, and then we can just start lining them up and go, here's all of our plants that are getting fly pollinated. So I started with the skunk cabbage, because it's also an Ericaceae. I started digging into the literature, and I also started having flashbacks about when I was a high school teacher in Portland, when I saw my first blooming skunk cabbage, which was horrible for the guy I was catching a ride with, because I saw them and I was like, holy crap, pull over. We have to go see that. And he's like, what's going on? Is there a bear? I'm like, no. See that field of like stinky stuff? I want to go out in that. And he's like, I don't want to be back in the car afterwards. Well, I go out there, I grab one, there's nothing blooming. I'm like, well, I want to take one home so I can look at it. So I go out there, I cut one with my knife, and I promptly jump back in the car. And we're getting, a mile or two down the road, car's vibrating. And all of a sudden, all of the pollinators start leaving. And they get so bad in the car, we had to pull over and take a 5-minute break while we depollinated the car. And so when you dig into the literature, it is not flies that are doing the pollination on the skunk cabbage. What it is are rove beetles, also things that love carrion. So I was kind of like, okay, you know what, fair enough. First one out of the gates, of course I'm going to be wrong. I'll start digging into other ones in this family, and we'll definitely find things that are fly pollinated. One of the cool things is skunk cabbage, if you ever get the chance to see it, do go up. I know it's going to sound funny. Go up and smell it. It is kind of amazing in the same way the corpse flower is amazing. And if you look at it too, I mean, it looks like the corpse flower, right? You've got the spathe, there's a spadix, and it gets all nice and showy, that bright yellow flower. And actually another fun kind of tidbit about this, we get skunk cabbage all the way down to Santa Cruz County in California. It gets really big and as you travel up the coast up towards Alaska. This is the plant that bears eat when they emerge from hibernation, because if you're hibernating all winter long, you have worked up the worst deuce of your life and you need some serious roughage to clean that system out. This is what kickstarts your first poo as a bear every spring, just FYI. Okay, so after the skunk cabbage, I was like, all right, let me jump into the other plants in the same family. I was like, oh, duckweeds. Wow, I would never have thought about them being related to corpse flower. Let me dig into the literature. Man, really hard to find any good papers about how these are pollinated. A, B, I didn't even know these things threw up flowers because I've looked at them. I've never seen flowers on them because, I mean, you really need like a little macro lens to get up there and look at them in the field to start with. So I was able to, they produce flowers. Then when you dig into literature, there's no hard literature on what they're being pollinated by. My gut feeling is they're probably getting a little bit of wind pollination just because you're on the surface of a pond, wind whips things across really well, but at the same time, you've got a lot of gnats and flies that are in the area. And I do find references from like Los Placitas College and a couple others stating that they have found flies with pollen grains from duckweeds on them. So potentially one of our native plants that is getting pollinated by flies, but really not super conclusive. And so now 2 species in, and I was like, okay, this is going to be a lot longer for me to put this together than I thought. And so I just want to put a new kind of asterisk on this PowerPoint talk. This is no longer the fly pollinated California native plants. It's now the fly pollinated California native plants, the asterisk edition. Here's the asterisks. From my literature search, there's a handful of studied Diptera only plants in California. And when I say a handful, we're talking a very, like maybe one hand. There's a handful of probably Diptera only plants, i.e. there's not a whole bunch of literature around it, but there's a lot of observational data and anecdotes. There is a handful of studied Diptera dominated plants that are what we call pollinator generalists. And these are kind of your plant that just is like, hey, everybody, I'm ready, come visit me. And all things from all sections of insects to animals come to visit them to then move their pollen around. Handful of those with good paperwork behind them. And then a lot of probably Diptera dominated generalist plants out there. And so in order to appropriately give you guys the ability to know when I'm talking out of my butt or actually when I'm talking from a reference material, when I'm talking from looser evidence, I made up this system for you. In the right-hand corner of these slides in the top, you'll have strong evidence, which is a stack of books. So, i.e. there is literature for this specific species dedicated to flies pollinating them as a dominant, or there is moderate evidence. That might just be, I found one paper or lots of papers around this genus, but not necessarily our specific California native that is fly dominated pollination going on. And then the last one, the guy just kind of sitting there thinking about it on the stinker pot about a fly. These ones, there appears to be lots of field evidence based upon anecdotes you can find online and even observational data from iNaturalist. So that's how to read the little cue card in the right-hand corner as we dive into the California native plants that are probably fly dominated when it comes to pollination. So when I started this, which was like Monday at noon, actually probably no, after I got back, which probably is not what my boss wants to hear from visiting Victoria. Boss, please don't be watching this on YouTube later. I started calling around all my botany nerds and basically saying, hey, tell me your favorite fly dominated plant. And it was just quiet. And I was like, oh, okay. All right, tell me your favorite plant you think probably flies like. And the one that rises to the top, if you're in coastal California or coastal Oregon, is the Fetid adder's tongue, which is a beautiful plant in the lily family. And if you've not seen this, you need to get off your couch next February and march yourself out to a redwood forest and start looking for these. These are some of the most striking lilies that are bizarre looking that we have in California, and they're not hard to find. But if you get close to them, you're like, wow, you're really pretty. You've got these really interesting decisions when it came to colors, these kind of more putrid-y purples and sometimes some red tones. And then if I get up in there for a whiff, woo, you are fetid. You're definitely fetid. And why is this? It's because fungus gnats are probably the dominant pollinator of these. Gnats, fungus gnats are a fly. They're in the order Diptera. And surprisingly, as you see the man sitting on the pot thinking about flies up in the right-hand corner, I cannot find a great resource, i.e. a scientific study that goes into the pollination of these. And so there's a lot of anecdotal evidence that fungus gnats are what's pollinating these. And you can see this in observational data in iNaturalist and all kinds of conversations online. The only paper I could find for this was in 2005, somebody at San Francisco State wanted to run GCMS, which is known as gas chromatography mass spectrometry, on the headspace. It basically means a fancy machine that takes a giant whiff of the flower and then can kick out what organic chemistry molecules are coming off of this that might attract things. But that is the best I could find in the literature search, surprisingly. So if anybody ever else finds better literature for any of this, send it to me. I'd love to update this PowerPoint or this slide deck with it. But once again, if you've not seen this, please, next spring, go out and find these. These are also, this is like one of the bell ringer plants that tells you, ooh, okay, spring is getting kick-started in coastal California. So it's another great reason to go out there and see these. All right, number 2. That was on the list of my botanist friends when I asked them about this, is another one where we don't have a lot of great paper evidence, academic evidence, but man, you talk to people that are into flowers and it's like, oh yeah, this one is fly dominated as far as pollination goes, because we even call it the stink bell, Fritillaria agrestis, which is a cool fritillaria. It's a rare plant in California and it occurs mainly in these large grassy fields. You can find it in Alameda County, all the way down, I think, to Slow. And there's a variant, another species very similar to it now, down near like the east side of Bakersfield. But this was my first year seeing it, so I was really excited to be able to talk about this one because I finally have some photographs of it. But it is one of these ones, you get up close to it and you take a big whiff, you're like, oh yeah. Nobody else except for flies would love this thing. But it's a really, really beautiful plant. It's in pasture lands, right, producing this stinky scent. So it's a place where you probably would have grazers around. You'd have lots of potential manure, things like that. So you'd already have flies in these areas. And while I was there trying to photograph these, I did see a couple of muscoid flies come through and take a peek at these. Another one, definitely no evidence for at all, except for a lot of anecdotal evidence now, is what I would say is maybe one of my top 5 California plants. And it's mainly because it's super rare and is hallmark California. It loves serpentine habitats that just look like Mars. You like look at these places, there are these giant talus slopes of serpentine, and you're like, geez, nothing would ever want to grow there except for this beauty lily family, Fritillaria falcata. And there's a couple of different populations kind of starting from about our area and then going down through San Benito County. It's a flower that's not seen very often because it is a rare plant. It's also in these really hard to get to areas. And this past year, I was lucky enough to kind of see it twice in 2 different areas, in Alameda County and down in San Benito. In San Benito, we went out at night to see whether or not, this is one of Damon's like sick weirdness things I like to do, is go out and look at plants with UV lights at night because they have crazy UV fluorescence, some of them. Falcata does not disappoint. So down the bottom corner there, you see that really bright red one. That's UV fluorescence at night. The exact same flower under white light is next to it. While doing that, we found a ton of these flies just chilling around these flowers. And if you smell them, they're also very musty smelling. So most likely a fly dominated pollinator because there's nothing else out on these slopes when it comes to insects when we're there during the day or at night. So super cool. Also, another great master's project if somebody wants it. Okay, finally, one with the triple book stack. We actually have literature for this one. We can say for sure. Brewer's bishop's cap, also known as Mitella breweri, in the saxifrage family. These guys are crazy looking. You'll find these in wet seeps in the western slope of the Sierra Nevada. These are one of the ones that for years I probably walked past them. And then when you finally realize that these exist, you'll be on your hands and knees every time you find a wet seep in California. Because you're like, oh, there's some of these there. They're absolutely gorgeous. So we don't have a lot of papers around our specific species, but there is just a thick deck of books around this genera having fly pollination in the form of fungus gnats. In fact, the literature is so deep that you can even find what compounds they are using to attract the fungus gnats. And so that is lilac alcohol and lilac aldehyde. And so you're like, oh, I'm familiar with that first part, the lilac part, right? Lilac plants, if you've ever been around them, you realize that at a distance, you enjoy them. They're visually nice. And at a distance, sometimes you get a nice kind of waft and it's like, ooh, lilacs. But if you ever get your face up into the inflorescence, it's a different story. It's kind of there's a little bit of funk in there. And that's due to these compounds, due to this alcohol and this aldehyde. And that is the same compounds that this plant is producing to attract fungus gnats in this area. One of my favorite plants in California, and this is the one that needs no asterisks on it, plenty of literature for this one, the monument plant. You find this up in the drier parts of the Sierra Nevada. It gets these giant inflorescences that are taller than I am. And if you get up close, they have just some of the most beautiful flowers I think you'll find in California. These are dominated by Dilophous pollination after native bees. So all of the studies that I've looked through for this, you'll find bees and this fly basically head to head as far as pollination goes for this plant. And there's a whole number of this genera in California, and they are some of the most stunning plants. I wish more people grew these in their gardens because A, they're super drought tolerant. They're really beautiful when they throw up their flowers. And even when they're not throwing up their flowers, the original leaf set is just this giant bouquet of green. And they're really hard to kill. So they'd be great for me. Another one that we can find a little bit closer to home for you Bay Area folks, there's literature on it. These are pollinated by local flies, especially hoverflies. And we have literature that points us to specific species for our Pacific false bindweed. This one you can find all over the East Bay. It's all over Marin County, going down to Slow County. This is a flower that you find all over the place. And for sure, the first couple times when I saw this, I thought they were being pollinated by native bees because I did a quick look and I didn't get close to what I thought those bees were, which were these hoverflies that have patterning very similar to some of our native bees, and they're about the same size. But once you get a little closer, you realize that they're dominated by the hoverflies around these, which is really, really cool. This is also another great garden plant for your backyard, drought tolerant. They flower after a lot of things are kind of done in California, when things are kind of getting brown and grass or brown and dried out, like you can see in the photographs there. And they're great for kind of using it as a tendril up stuff. Another one, there's a lot of great literature for that is fly dominated, bees aren't involved, are basically Ipomopsis aggregata, the scarlet gilia, which is another great native plant for gardens. They have these great inflorescences. They're relatively quick is the only bummer about them. The flowers are up for about a week and a half usually, and then that stalk will go down and it'll usually shoot another one, but they are just absolutely gorgeous. And if you find these in the wild, you do see it. They have just cifrid flies over them all of the time. These I predominantly find along kind of the Sierra Nevada ridge where you're getting started that dry habitat. Close to home, mule's ears, they definitely are getting pollinated by our bumblebees and our local smaller bees, but they also have a lot of fly pollinators. In fact, there's one that half the reason I wanted to put this in the presentation was this specific fly that has a horrible name, the sunflower maggot. It is a really cute and attractive fly. It is one of the easiest flies to observe that I've ever seen in my entire life, because if you're up, say like in Sibley or one of our local parks to Oakland, looking at mule's ears in springtime, you will find these easily in the inflorescence or on the leaves. And if you find them on the leaves, the funny thing is most flies, as soon as your hand is close, they are gone. This one, you can literally take the leaf, pull it back, and it will just look at you. And then it will kind of walk a little bit. And if you go in there with your hand, I'm that type of person, like, you know, like, well, I wonder how close I can get. You can literally just kind of escort it around the leaf with your finger. And this thing only occurs in our area on mule's ear. All the observations you'll find on iNaturalist, anybody you talk to, this is the only plant you find it on, which is pretty cool. And when you find them a lot of times, like my photograph here on the right, you'll find that they usually have pollen grains on them. And so they are definitely involved in pollination of mule's ear, but we just don't know to what level they are doing the pollination. Some of my favorite plants in California, as far as an edible point of view goes, are in the rose family. One of the most striking ones that I love, even just for the flower, because the fruits are kind of tart, is the salmon berry, Rubus spectabilis. These have these just beautiful purple flowers. And like that earlier study that I showed you that was in the Smithsonian magazine based upon 2019 work of crops in the United States, we know that most rubus are fly dominated. And so when I say rubus, we're talking everything that looks like a blackberry or a raspberry. So this is one of them. This you can find, I think, down to Santa Cruz County, but once you get up to like Del Norte and Humboldt, this is one of the dominant kind of brambles that you will find in the forest there. Absolutely gorgeous. The fruit's a little tart even when it does get ripe. And if you dig through iNat, you will find plenty of these with flies hanging out next to them. My favorites, however, though, berry in California is the thimbleberry, Rubus parviflorus, also in the rose family. And the reason I love this is for a number of reasons. I think the fruit is one of the most delectable fruits we have in California. You'll never see it ever make its way to a supermarket because someone would have to do a lot of genetic engineering to get this to hold together long enough in its ripe state to ever make it to somebody's mouth. You literally have to be at the plant at the right time to basically pull this off and get to taste them. They're absolutely wonderful. The other reason I love this is they have just one of the largest flowers when we talk about rubus in California. Just this giant spray of petals, like, hey, here I am. And the leaves, you just want to like rub them on your face. They're just some of the softest leaves you will ever find. They make these nice kind of large, almost like hedgy looking growths in area. They don't stab you like the rest of this group does. Also another one in the rose family that gets a lot of cyprid action is cinquefoils. And so a lot of cinquefoils we find across California have a lot of cyphrids on them. And this is another one where a lot of anecdotal evidence that these are fly dominated, which the more I look at roses, the more I'm thinking roses were really for flies. One of the ones I really like in the East Bay that we find in large swaths that is easily missed unless you're taking your time to look at our wildflowers is the distant scorpionweed, Phacelia distans. And the reason that it gets like overshadowed a lot of times is because you'll see kind of stands of it and it just kind of looks like a scraggly flower until you get up close and you look at the petals, and the petals are just amazingly intricate. And while you're there, you'll realize that there are lots of different flies coming in and out of this and very few other things. You'll see a couple bumblebees attempt it, but usually the way that these plants are up on a stalk, as soon as a bumblebee gets on it, it just kind of like falls over. So they don't get much of a chance to be on there. There's a little bit of evidence that these are mainly fly pollinated, this specific species. However, though, we have one that is for sure flies do the big business when it comes to pollination, which is our quote unquote blue tansy, Phacelia, and I'm going to butcher the Latin on this one, Tanacetifolia. This one you'll find as you get a little bit further south or a little further east from the Bay Area into really dry habitat. So these photographs are ones I took down the Carrizo Plain a couple years ago, where this one can just be gangbusters, right? Entire hillsides of them. But tons of evidence that this is a major cyprid habitat and pollinated by cyprid flies. And so it's interesting to see this one that has gotten so much study because people are actually talking about this as potentially something you would plant by crops in order to facilitate more cyprid flies being around because they're a good habitat, they get pollinated by them, and they can potentially spill over to your crops. and cause a little bit of knockdown on the aphids too, which is good. One that is one of our largest kind of generalists in California when it comes to getting pollination, anything in Apiaceae, anything in the carrot family are just the plants that are like, hey world, I'm ready for you. Everybody come check out my blooms. And you definitely see this with Angelica linear loba or Sierra Angelica, which you'll find just in the Sierra. You will see this from like half a mile away sometimes because they love to be in giant exposed areas and they'll just throw up these huge clusters of white flowers. And when you go up to these flowers, they are just the hotbed of insects. Every insect you want to see is going to be there, except for maybe some of the moths, unless you're hanging out there at night. Tons of butterflies, tons of flies, tons of bees, tons of wasps. Everybody's there. It is the disco. However, though, if you dig into the literature, not for our Angelica, but for a very closely related Angelica in Europe, there's a study that just came out 2 years ago that shows, despite that it is the disco for the insects, it appears to be the flies that are actually the ones facilitating the pollination. It's not the bees, it's not the wasps, it's not those freeloader butterflies that everybody loves. It is the flies that are really moving the pollen around, which is kind of cool. And if you ever see these in California, give yourself 15 minutes and just hang out by one and see who comes by. You will find some of the fanciest looking flies of your life, like this disco green one that's at the bottom of this slide. I think it showed up maybe 4 or 5 minutes into me watching it, and I was like, oh, I'm so glad I just decided to take my water break by this plant. No talk would be complete unless we couldn't hit an orchid in California, right? So this is definitely loose hit, because this orchid is really well known for being self-pollinated, but there's also a lot of evidence that Calorhiza, the coral root orchids, also have some pollination that takes place due to some ephesus flies. And so there is a paper that shows our spotted coral root orchid does get pollen transferred from one set of these flowers to another. If you've not seen these, this is another one that should be on your to-do list in California because they are everywhere in our forests. They're a mycoheterotroph that loves sucking the life out of Russula mushrooms. And so if you know where Russulas are in the wintertime, go back there during the spring and you'll usually find great stands of coral roots all around. There's a ton of these in the Oakland Hills. They should be blooming right about now. We've been looking predominantly at like lower story stuff, kind of herbaceous plants. So I was like, before we end this, I've got to get to some canopy looking stuff. Anybody that's ever interacted with dogwood, you know that it is a beautiful flowering tree. You also know that if you put your sniffer up in it, you immediately regret it because they do not smell nice. And so you might scratch your head and be like, well, who the heck is coming by to pollinate it? Flies. Plenty of literature out there, not for our specific nuttuli species, but for other species that flies come through and do a major amount of the pollination for these flowering trees. And the other one, just to kind of hit, last plant of the night, is a Ceanothus. And that's mainly because Ceanothus are one of those hallmark California plants. They're also one of the most awesome plants to put in your garden because they're easy to take care of. Pollinators love them. They are gorgeous. They also make a nice cocktail for you cocktail enjoyers. I love making cocktails out of some of the blooms. But these, there is good literature that some of our Ceanothus in California, like this native rare one, the Lakeside Ceanothus that you see down by San Diego, is fly dominated when it comes to pollination. The ones I see up here in Northern California seem to be getting an even mix in the field when I'm out there looking at them. Our native bees love them, our native flies love them, and even a handful of our kind of wasps will come through and enjoy them.
Look at it. It's
Tying this up, our native plants depend on diversity of pollinators. So when you hear the word pollinator, don't always think just bee. Think fly, even get out of the fly zone, think beetles, think ants. There's a whole bunch of people involved in this. And remember, some plants need very, very specific pollinators, right? They've evolved maybe to have a partnership with only one pollinator. And so making sure that we provide habitats that allow those pollinators to exist also mean that those native plants that we love get to exist. And so one of the things I wanted to highlight that I've been seeing going on the last couple of years, and actually Pollinator Posse has done a fantastic job at this, which is taking some of this call, this call to action you see in a lot of popular media of like, oh my God, save the bees. When people say this, they're like, save the European bees, which in my head is like analogous to saying, oh my God, save the cows that we brought over, right? These are an agricultural organism that we brought over that does give us a lot of benefit. They are great pollinators. We get that sweet stuff out of them, which makes us love them even more. But even more than just saving these, an agricultural tool that gets us a product, the conversation has started to shift, thanks to groups like Pollinator Posse, about what can we do to encourage saving the rest of the pollinators, our native bees that are super diverse in California, over 1,600 native species. And the interactions with these and our local plants is just starting to be known. I saw Terry shake her head. I probably got that number wrong. It's probably closer to like 2,000 at this point, or 2,200. But like I said, we're just starting to scratch the surface on understanding these systems, let alone just being able to name them and then understand the interactions. And then what I foresee coming in our future is not only is it going to be save the native bees, we'll even take this one step forward and start thinking about how to expand the love to the other pollinators. And this will happen. I don't expect this to happen in the next year, 5 years, but maybe towards the end of my life, we'll start to have a grander appreciation for how do we get to a point where we make habitats and ensure that the habitats exist for our other pollinators to exist, because this is the only way that we still get to enjoy a lot of our native plants that evolved to only work with those native pollinators. And just to throw it out there, a couple things to be aware of that are coming up on the calendar. A talk worth attending, June 23rd, 7:30 p.m. California Native Plant Society's East Bay chapter is having this really important talk on the insect apocalypse. So if you've been looking at the literature, there's been a number of studies that come up the last couple of years, longitudinal studies showing large drop-offs in flying insects. This has major implications on our ecosystems, which then has major implications on our food safety, which then has implications on economic systems and geopolitical behaviors. And so I would say attend this talk to at least get a foundation of what's going on there, because all of us being involved in figuring out how to encourage pollinator habitat is maybe what helps us skirt an insect apocalypse that leads to geopolitical problems. How do we do this? By saving habitats, rebuilding habitats. So thank you, Pollinator Posse, for being one of those groups that really does the education where it comes to rebuilding habitats. And depending upon where you live, there's a lot of groups that are interested in saving those natural habitats. Look for them, find out how to donate time or money to them, because these are the two things that are going to allow us to maybe skirt the insect apocalypse. With that being said, I want to say big shout out to Pollinator Posse for what they do, and to these 3 other groups I leaned upon for this talk, which is Insect Science Museum, which I've learned a lot from the past, Holy H2O, which is mainly watershed focused, but does a lot when it comes to understanding and documenting local insects. And then of course, my go-to platform for documenting and peeping in on what other people see, iNaturalist. With that being said, thank you guys for putting up with me for that long. We'll open it up for questions.
All right. Can everyone just unmute themselves? And Damon, I want to say thank you so much. That was amazing. And most of the comments in the chat were that this was a really good talk. And thank you. And you're amazing. I want to thank from the Pollinator Posse, from the bottom of our hearts, Kyle and Susan for turning this into a Pollinator Posse fundraiser. Everyone can still go to pollinatorposse.org if you would like to donate. You know, where the money that we generate usually goes into pollinator plants and seed, and we're just trying to get it out there. I do want to say one thing about Damon. What you said was fantastic. It was absolutely fantastic, but not just to get more native California plants in the ground. What's super important right now, and that the posse is very focused on, is that know where your plants come from. Make sure that you do not get, because some of these, the honeybees are much stronger against pesticides than the native bees. And that's what we're finding out is our local native bees and flies and stuff are super susceptible to the neonicotinoids and all the plants that they're spraying at big box store nurseries. And, you know, if you can, you know, buy them from your local CNPS, that's really the best. But that's like what you can do as consumers is don't buy the pesticide ridden because you know it says bee butterfly friendly, but you get it home and it's killing the bees and the butterflies. So that's super critical when you're out there getting these native plants. Thank you so much, Damon. Terry, do you have any closing remarks?
No, I learned a lot. Now I've got to go modify my talk to add the flies in.
Yeah, Damon, we would love to do this again, you know, and do some even deeper dives about, you know, the compounds that we really need to look for in the native plants and our local right here in Lake Merritt. What specific plants I'd love to work with you about right here in this area that we need for those bees that only have one particular plant that they can pollinate. And, you know, those kind of really specific things would be really fun to help our neighborhood, because that's the biggest question we get at the posse is, I just want you to tell me what plants to plant. I mean, you don't want to do the research, you know? So I think
Right,
that's the biggest benefit for this community.
And that's an important thing to bring up is that, I mean, there's some times where we do know, right? There is so much we don't know about our local ecosystems. And we can all be involved in figuring some of this out by just observing and documenting. And like, that's why I tell a lot of people use iNaturalist, just because it's a nice clearinghouse to kind of put some of this, but also contacting your local CNPS, California Native Plant Society, or other kind of gardening groups that are very natural or native plant focused, and working with other people, right? This shouldn't just be you on your own. And I think that's what, one of the things that you feel in your heart most when something like this happens, like when everybody ended up in Kai and Susan's backyard looking at the corpse flower, the number of people that were just having conversations about other things was just incredible. And so this is a community thing. Don't feel that you have to do this on your own. What I would say is, first, find other people around you that are also interested in this and get together with them. And then figure out how can you monitor what's going on, how can you make good decisions about what plants to bring in and what habitats to build. Because not all the answers are out there, and we will find them together. And there are a lot of resources out there, though, that being said, but there's a lot we don't know too.
Right. That's why that's why we named ourselves the Posse, because we knew we couldn't do this alone, that we were going to have to work with other agencies and other entities. And, you know, we wanted to build a community around conserving pollinator habitat, but that's actually just the local ecosystems. Because if you put your hand in the ground, you're affecting the ecosystem, period. So thank you, Damon. It was awesome. I don't see, there's incredible talk. Thank you so much, Damon. Loved the talk. I learned so much. Thank you, Damon. Also, Terry, thanks for explaining everything to us when we were in the greenhouse. So it's all really just really super positive. So, you know, there's really no questions. So,
and let me do this. If there is a question, because like this is the way my brain works. Like I watch one of these.
oh,
and I'm
here's
like,
one.
I burned out at the end of it. If you have a question that comes up later, there's my email address. You can just shoot me an email, and I will get back to you with if I know it, or I'll probably spend my night looking for it and then get back to you.
There's one question.
That question that comes up later. Yes, go for it.
There's one question. They want to know what kind of fly landed on Pence's head.
Oh man, I don't have a picture of it in front of me, but I think it was just a domestic housefly, which is usually, it likes, you know, rotting things. Which, you know, if you're under a lot of stress, you do produce a lot of those compounds that is similar to rotting stuff. So,
right.
No poop on Pence per se, but.
So I have a question, Damon. Our pipevine are pollinated by fungus gnats. So that, I thought you mentioned that that's fly family as well, right?
Yep. And so they do make it to those, yeah.
And now we noticed a new fly that was eating the pipevine at Terry's house. They were eating the eggs. of the pipeline
Sitting there
swallowtails.
sucking them out. I have a picture of it. I should send it to you. I tried posting it on iNaturalist and it didn't, nobody got closer than Diptera.
Just if you do at Damon Tighe on iNaturalist, that pops up into my comment field and I'll find it and look at it. Or you can email it to me.
Yeah.
Yeah, we never noticed that before.
Yep. Well, it's amazing, especially I think with a lot of biology. We forget that a lot of biology happens at a small level, that it takes patience and looking, like determined, conscious looking to then notice these things. I mean, that's one of the biggest benefits, I think, of the pandemic time. A lot of us spent a lot more time locally, right? And I've lived in downtown Oakland since April 2004. I feel the number of species I saw this past year just exploded my brain. Because I had more time dedicated to be around home and to actually go out and consciously be, you know what, I need to take a break. And the way I'm going to take my break is going out and looking at things. And you start finding things. So build time in your schedules to go out and just look in your local habitat. And sometimes that's like hard. Like it's hard for me to like say, I'm going to go out and sit by Lake Merritt for 10 minutes and just look at a few things. But all of a sudden you start to see new stuff because of that time for yourself to do that. And it's also really great for your brain.
So we'll make sure that we post your upcoming talks, Damon, to these people, and please let us know, you know, at the posse, we can put it under our events. Thank you so much. And thank you, Kyle and Susan and Terry. And please go to pollinatorposse.org. We do have under resources, we have a plant list that attract pollinators. We want to make it as easy for you as possible. And we'll soon be putting a list of nurseries on there that are neonicotinoid free, so that it won't be so hard to find the native plants. Because that's really a really big mission we need to focus on, and be, you know, because the consumer controls the retail. So if we stop, you know, we stop buying it, we start demanding neonicotinoid free plants, that's going to make a difference. So thank you everyone for coming. This was amazing. Kyle and Susan, can't wait for her to bloom again. All right. Thank you, Damon. It was amazing.
Thank you.
Thanks.
Good evening, everybody. Bye.